Okay, good morning. I already had one question about the homework from yesterday. Any others before I launch into new stuff? Yeah.
For V1 values, I get different answers every time I ran my V1 values. Okay.
For the gradient descent, you mean?
Yeah.
Yeah, that doesn't surprise me that much. So, yeah, the Amazon Web Services thing being down yesterday messed me up probably as much as y'all in terms of what we were going to do today. Part of the lesson that I hope you get from that homework assignment is if gradient descent doesn't converge, you don't get the right slope, okay? But you don't know what the right slope is when you're doing real analysis, right? If you've got big data and whatever software you're using defaults to gradient descent to estimate the relationship, you don't know if it converged unless you look. And so that plot of the iterations versus mean square error and whether that has reached convergence, that's what you need to know whether your model has sort of reached an equilibrium. And this is just one example that will be true. We're gonna do PCA all day today, but it's very important that you look at the data and look at what the algorithms are doing. And I bring this up because it's back to the beginning of the semester, and how do you use ChatGPT? You have to have a test and write the test before you do the analysis. So in what we've been doing lately, I haven't made this very explicit, but what's the test? How do you know if any of the code I gave you is giving you reasonable answers? If you're just trusting ChatGPT, that's not it. I'm losing in that case. How do you know if the code for ordinary least squares regression actually did least squares regression? Well, one is you could ask ChatGPT to read you the code and what it did, but the test is written in there. I'm trying to connect some dots here. What is the test? And how can you test yourself on new analyses or test your code? Yeah. And so how have we done that? Like in I think every example that I've given for Colab, what's the first thing it does? Yeah, it simulates some data, right? So you know the relationships that are embedded in that data. And then you do the analysis on that data, and hopefully it recapitulates the input parameters. In the regression, it's easy, right? You gave it the slope to simulate the data on, and then you run the regression, and ordinary least squares gives you that slope, okay? Gradient descent in the homework, the reason I told you to slow it down so that it basically doesn't converge is so that you see, back to your question, it doesn't get to the answer. And you know it's not getting to the answer because you simulated the data. You know what the answer should be. And that's relevant for, like, if it was really a big data set and it was something that you couldn't or wouldn't want to wait on ordinary least squares to transpose those matrices, you don't know the ordinary least squares sort of right answer. You just know the gradient descent answer. Okay, but is it any good? Okay. Okay, hopefully that connects some dots, and when you go off to do, starting next week, independent projects, I am going to want to see how did you test whether your analysis is doing what you think it does or is supposed to do. All right, so week 10. This is week 10. It's module 10 anyway. I updated the PowerPoint, so it's the same PowerPoint, but you want to re-download it just because I added a couple slides there. I thought I had the unsupervised learning approaches on the slide before last week, but when I looked at it yesterday, it wasn't there. So this new version has the unsupervised learning approaches on it. And then the Colab notebook is what we will work through for the rest of the day. So we talked about supervised learning a little bit. What characterizes supervised learning? See if you remember. It's in the slide before that, but what is supervised learning? And what, in the sense of what's different between supervised and unsupervised? Yeah, labels, that's the difference. Or targets, sometimes you hear it called targets. In the sense of regression, what are the labels or the targets, right? Why is regression a supervised learning approach? Well, you have the X and the Y data though. So what are the labels, right? Why is it supervised? The Y are the labels for X variables, right? So it's your dependent and your independent variable in the classic regression sense. So the X is hopefully telling you something about the Y values. You know both of them in this case, and so you're able to predict a known Y from an X. Ideally, that's what the regression is at least telling you on average. So unsupervised learning approaches. This is when you don't have labels. So all of these are ones that you'll see pretty often in the literature. We're going to cover PCA for most of today, and then at the end talk very briefly about TSNE and UMAP, which are, so principal components preserves the variance, assumes linearity. TSNE and UMAP are mainly just for visualization, but they're nonlinear. They're taking sort of complex multidimensional data and trying to put it in 2 or 3 dimensions so that you can see the structure in the data. Okay. And that's sort of stepping us to classifiers, which is what we're going to do on Thursday. And so how do you train the machine to know what a mug is, or what a T cell versus a B cell is? Okay. When we talk about PCA, just one bit of jargon that comes up and sort of math to demystify what's going on behind PCA. But PCA assumes there are some latent factors. These are the variables that you didn't measure, but you're trying to infer from your data. So they're called latent just because they're hidden, like you didn't measure them. And the only reason we know they're there is that there are patterns in the data that we did measure. You can think of it like genes that are co-expressed are part of a pathway, right? And so maybe the better representation, instead of taking any individual gene expressions, is to take the output of that pathway, which would be a principal component representing all those correlated genes that are being expressed. Just as one potential example. Really, ultimately, those latent factors are because there is some combination in your measured variables that are correlated. A little bit more formally, right? This looks like this equation as a multivariate regression, but that's a regression for one of your actual measured variables. And so what this is saying, we've measured x1 through xp. That p is our number of features, or our number of measured variables. For each one of those, we might assume that there's some latent variable that explains that. So in this case, say our first x variable is going to have a combination of weights based on those hidden variables that are actually determining the variable, the value of gene 1, for example, right? I'll come back to the TSNE and lab notebook. I'm curious, people that are in biostats or biometry this semester, have you already done principal components? No? Actually don't know if they get that far or not. Okay. Whoops, that's not the right notebook. So I zoomed through this at the end on Thursday. Just gonna go back a little bit to say that the idea behind principal component analysis is somewhat similar to regression, except that you don't have the y variable. So you are only looking at which of my x variables has the most variance, and then basically trying to account for that variance in x. You can think of it like the residual after you did the regression line, and then you ask then, of what's left in my residuals, which x variable explains the most of that variance, okay? And so you sort of without actually doing the math, it's hard for me to explain it much better. The best way in terms of efficiency is you can ask ChatGPT, what does he mean by PCA is computed by eigen decomposition and singular value decomposition? Or it should be or, okay? Just two approaches. Oh, I did change it to or. This is the terminology that you often see, which is probably why ChatGPT, when I ask what's it doing, it says it's rotating the data. What does that actually mean? Okay, so let's run the cells and then we'll talk about what's going on. So we're gonna first generate a data set, 200 samples. It's got mean 0, standard deviation of 1. And then we've got, that's the latent variable, z. And then we have two x variables that have slightly an x and a y that have slightly different amounts of variation in them, okay? And then here, unlike before where we just simulated y, here I'm simulating the actual x value that'll be used as a function of the latent variable z and this EPS x. Right, so we know our true slope is 1.2 and our true intercept is 3. So that's the data. Before we actually look at pictures, if you just do the regular ordinary least squares, that's your slope and your intercept. Again, how come they're not 1.2 and 3? Right, so my slope, I simulated the data at 1.2 with an intercept of 3. How come ordinary least squares didn't return that? Is it broken? Is my code bad? It's not rounding. Well, I mean, if I round to those, then I get to the right answer. But that, I mean, the precision is relevant here. How come it didn't? I asked the same question on last Thursday, and you guys got it right. Were you even the same one that said error last time? I think so, yeah. Right, because we've simulated error. And in this case, we even simulated a latent variable that has some forcing on x, okay? And so PCA1 is if we did the regression on the principal component rather than the x variable itself. So if we did it on z versus the, not on z, but now let's plot it, see what it looks like. So you can see those equations from the last one plotted here, and they're slightly off, right? Because remember, drive this point home multiple times, the PCA doesn't know what y is. It's only looking at what's the axis of most variation in x. And one of the things that I went over really fast on Thursday was the fact that if you look at the residual, and this is why I sort of hedge to say, you can think of sort of the second PC as trying to explain the residual, but it's not the residual of the regression per se, because the residual of the regression are these blue dotted lines that are in the vertical plane. So it's like, how far off is the line from the expected y value, or from the observed y value, because the line is the expected y value. Whereas the principal component is just saying how far away is each dot relative to some estimate of the mean in that range of the x. So it's orthogonal to that orange striped line. if you look at the error, so if you took all of those differences, but right, all the sort of orange or red differences and the blue differences, you would see that the mean square error here, so that's the one computed from ordinary least squares, is a little bit more than the orthogonal distances. Yep.
Are these metrics comparable though? Because they're on different, like, you don't know if they're the same units.
Well, they are on the same units.
But then isn't MSE on like the y axis, like values, and then this is based off of the x?
They're both estimates of it's the x or the y minus the expected value squared, right? The sum of those values. So they are more or less on the same scale to the expected value for either, say, that orange line representing the principal component or the blue line represent the regression with y. I could be wrong, and I'm going to bring up the root mean square error in a minute because data science program is doing its job. Zanib. So that's good. I'm pretty sure these are on the at least comparable. We'll talk about scaling when we get to the RMSE versus MSE in a second.
Does PCA do like scaling, like standard scaling automatically? I personally have never used it, so.
It scales everything to zero, mean zero, like
Including
all the
target.
variables.
Including the target.
There's no target.
Right. Okay. Thank you.
Mm-hmm. Good questions. Those, I mean, these are the questions that you should be thinking about. Okay, so if the mean square error for, or mean orthogonal distance, they're the same in my head, is less for the PCA approach, why not just use that instead of regular regression? So this is the question here. Why not use PC1 for this model, right? So instead of having our, actually this, man, that's a typo. Right, why not have PC1 times some weight plus the error instead of y equals x plus b1 plus error, right? So w and b1 are the same, right? It's the slope. Any intuitions? So if you run the next cell, it will give you both of those models. It will run y being predicted by x and y being predicted by PC1, and you see that, well, somebody interpret that for me. What does that tell you? My confusion last week, this is RMSE, this is where I left it both RMSE and MSE later. It's the square root of the MSE just puts it back on the same scale as the data. So remember, mean square error takes the difference between the observed value and the mean and then squares it so it loses directionality so that you can sum it up, right? And so having a large sum means all your observations are far away from the mean, okay? But then since you've lost the directionality when you did the sum, that is no longer really on the same scale as the original data. When you take the square root of that, it puts it back into a meaningful scale relative to the original data. So here, I could put MSE and like that distinction. Zaneve and I had this conversation after. They're totally lost in my mind because I'm so used to just comparing between models. I can compare models on an MSE scale or an RMSE scale. The biological interpretation, though, is more meaningful on the RMSE scale because it tells me how many units, right? So we know, I forget, what did we put this range on? I have to go up and look at the, oh, it's a mean and it ranges plus to minus 1. So, not plus to minus 1. It's still plus to minus 3, it looks like. Doesn't matter too much. Okay, back to my original question. Why not use this one and asking like, how do you interpret the difference between these two? Which of those models is better? Talked about how R squared and MSE are the two things that you look at to evaluate your model. Yeah. So it seems like PC1 is the better model. The R squared, right? That's how much of the variance is being explained, and it has lower residual error. Okay, that's the RMSE. The issue is that in this data, X is a pretty good predictor of Y, which we know because we simulated it. But even if you just plot X versus Y, you see there's a pretty good relationship there. And so if you only have the variance in X to explain Y, then doing that principal component analysis might tell you more about the relationship with Y because it's the only thing that can explain it. But it's not guaranteed to do well at all on new data. So this is tricky, and the next few cells are meant to illustrate this when we expand it to more than one X variable. But there's an important lesson in having a good model versus having a good predictor that's going to become relevant when we talk about classifiers. So just because the model fits the data well doesn't mean it's a good predictor of new data. Okay. All right, so I'll run the next cell here. And this is going to do a few things. It simulates two X variables, and then does the regressions. And so the first panel here is showing you X1 versus X2. Somebody tell me what they see. What's true of our data?
The two X variables seem to be correlated.
Yeah. So X1 and X2, I mean, you could do the regression of those two and probably get a good result, or the correlation, right? They're correlated. And so when you do principal component analysis on these two, so, right, we try to figure out if there are latent variables that explain the variation in these two X variables. What we see is PC1, so now this has the principal component value on the x-axis and the y on the y-axis. The principal component explains y pretty well. Principal component 2 doesn't really explain y. There's almost no variation left in x. Okay, so second set of panels here shows you x1 versus y. So if we actually did two separate regressions, x1 versus y and x2 versus y, somebody, what would your take-home message be here in terms of which one is a better predictor? I heard whispering. X2 looks like a better predictor. Why do you say that? Yeah, that's the sort of concrete indicator. The R square for X2 is 98.98 versus 0.79 for X1. And then, like, visually, right, there's just less variation around the line. X2 is going to be a better predictor. Now, if you go down, I've had it print out the loading. So this is what you often want to look at in terms of trying to interpret what your principal components are. And this is a toy example. We've got two X variables and I'm simulating and I'm estimating two principal components, right? So not something you would normally necessarily do, but I'm trying to make a point that we'll get to in a second. But which principal component corresponds with which X variable here? For the most part. So how you read the loadings table here is that is like higher values mean more of the variation in that X variable is explained by that principal component. Okay, so high values, and it can be plus or minus, right? You can have a really high negative number and it just means that it's influenced in the opposite direction of other variables on that principal component, right? So 0 would be this X variable is not controlled by this principal component at all. High positive means that the directionality is somewhat arbitrary, but you can think of it as a dot that lies above that line. It gets complicated when we start to expand this to multiple dimensions, but high numbers, high loading means that principal component and that X variable are more correlated with each other, okay? So, back to the question, which X variable is more associated with principal component 1 versus 2? Sorry, I can't hear you, Oscar.
Oh, X1 for PC1 and X2 for PC2.
Yeah, X1 is basically PC1 and X2 is basically PC2. Now, there's some influence of X2 on PC1 and vice versa. PC X1 on PC2. But for the most part, that's true. And so if you did a principal component regression, right, most of, actually, let's just, most of Y would be explained by PC1, even though we just, like, X2 explains Y better, okay? And that's because principal component analysis doesn't know about Y, okay? Going back to, like, what's the point here? If this seems a little confusing, that's okay, but much like the gradient descent that I talked about at the beginning, I said I'd come back to look at your data, right? So it's important that you understand what's going on with your data. In this toy example, which you would never do a 2 PCA on 2 variables, but the thing to notice is that, like, these 2 X variables are explaining the same thing, okay? And so you would probably just do the either the regression on PC1 alone or the multivariate and just leave both of the X variables in there. Anybody, so have you guys talked about multivariate regression in stats? You guys heard the term multicollinearity before? Yeah. The danger with doing the multivariate with both X's in here is that since they're explaining the same variation, this is an example where when you've got correlated X variables, they call that multicollinearity, and even though your model may fit well, it is more likely to be a bad predictor of new data, okay? I'm not going to unpack that, but let's go to the multiple, let add some variables, and some of this will hopefully become more clear. Can't promise clear, just more clear. So here, now we're going to simulate 5 variables. Sorry, not 5 variables. Yeah, well, the columns here, 1, 2, 3, 4, 5, and then enzyme activity is the thing we're trying to predict. Put real names on them instead of just numbers, so that, well, in the hopes that having something concrete will help with the understanding. So first thing, we'll just run multiple regression on those variables. And I'll encourage, just go back later and look at these are still simulated data, even though I put meaningful biological names on them, still simulated. It's not real data. So in principle, we know what the right answers should be. So if we do a 5-way regression, right? So we've got X1, 2, 3, 4, 5. each with their own slope. So this is what it's telling you, the intercept, and then the weights or the betas, the slopes for each of the variables. In multivariate regression, right, the sign only tells you the direction. The magnitude tells you something about its influence. So higher magnitude means it's has a stronger association with y. Okay, and so this is where I meant to point out the MSE, RMSE thing. I left them both in there this time. Okay, so on the scale of enzyme activity, 0.8 is on that scale. This is just the variance estimator for it. But our model with all 5 of them in there explains 94% of the variance, right? R squared's pretty good. If you wanted to visualize that, right, is this like how, this is where it gets complicated, right? How do you do 5 variables on one graph? We can do 4. We'll show 4 in a minute, but if you were just staring at these, like, you can learn something. Before we go run some more cells, which variables would you guess are good predictors or the best predictors of enzyme activity? Why do you say that? The answer is yes, maybe.
Like I know why in my head, but I'm trying to figure out how to word it. So like, that's where I'm getting stuck. Because I understand, but it's hard to put into words. I'm confused.
What's the intuition?
Well, like, just looking at the raw data, I saw that there was, whenever gene A changed the most, that was when we saw the most significant influence with the enzymatic activity versus the other raw data.
Good. You're definitely on the right track. So one way, just staring at this data and this is what you should do when you've got your own real data and you don't know what it means. Stare at the data for a while. Okay? So you start to see here that gene A, for example, when it's in there, you have a steeper slope in many cases. It's hard to see in that one, but especially when you've got A and B here, notice the y-axis. The y-axis isn't the same in all these graphs. So there is a much steeper slope indicating larger effect of this AB pair on enzyme activity than there are in any of the other ones. And right, if I was going to go back and do this on my own data, just as I'm staring at this data, I would say, oh, these aren't all on the same axis, so I should put them on the same axis so that I can compare. And what you would then see is this one, even though this looks like it's pretty well sloped for stress marker and metabolic, if I put that on the 20-point scale, it's going to look flat. Okay. So some indicators there that, maybe gene A and B are important to enzyme activity, or at least more important than some of the other ones. But let's keep going. Let's we've got 5 variables now. Compute the PCA. And I should say, I don't have it marked in here, but you can note to self, whatever. I've limited this PCA analysis to just grab the first 2 components. It'll grab more later. But this n components, you can say none and it will do as many as you have measured variables, but just letting you know where the 2 PCs come from. Okay. PC1 and PC2. PC1 explained 44% of the variance. PC2, 25% of the variance, and that's across the X variables, right? If we plot those first 2 PCs together, so this is often called a biplot, where we've got our 2 principal components and then it's color-coded by enzyme activity. So the value of enzyme activity. Somebody interpret this just in terms of the principal components. So like, I don't want you to necessarily have to put function on them because we haven't talked about the loading of the different X variables on these principal components yet. Just asking you to read this graph. Like, what leads to high enzyme activity?
Largely higher PC1, whatever variables are linked to that principal component are directly linked toward enzyme activity.
Yeah, excellent, right? The main spectrum of color is across PC1, right? A little bit, you know, the brightest yellow dot is a little bit high on PC2, so maybe there's a little bit explained by PC2. But then exactly what you said, so what we would want to know then, which variables are important for PC1, because that's what's really determining enzyme activity, okay? So this is just a fancier way to plot that data. This plot and this plot are the same thing. It's just instead of color coding, I've now put a z-axis on there. And, right, so if I spin this around, I'm not a huge fan of how Plotly keeps flipping the background, but, and you can see, I think I left the color coding in here, yeah. So if you wanted to really do that, you can, in the figure plotting, there's a marker note that is commented out. So if you just switch which one's commented out, you can see it really is just like the other one. Okay. Of course, that's switched around. Anyway, just showing you alternate ways to look at the data. If I'm going to keep harping on look at your data, show you meaningful ways to do it. Okay, so then the next cell is running the regression of those 2 principal components to predict Y. So this is like a 2-factor regression with just PC1 and PC2 versus the 5-way multivariate. So you've got 5 X variables trying to predict Y. So the full 5-feature explains 94. We ran that first up at the top, so that's the same. Nothing's changed. If you do it on just the first 2 principal components, you get to explain 83% of the variance, slightly higher. So the 2 principal component is not as good, right? in this case, maybe you would still run that full multivariate with 5. Next cell just showing you the loadings there. So again, going back to this question of what do interpretation biologically. What explains most of PC1, which in turn explains most of enzyme activity? Gene A explains most, closely followed by gene B. Okay, so this is kind of just putting numbers on what we noticed in the two-way regression plots that we ran at first. Also, stress markers also it's not that much lower of a number. It's just in the other direction, right? So that's kind of interesting in terms of potential biology. And then for PC2, you can see like signal protein level and metabolite concentration load pretty highly on that. But then keep in mind that the explained variance is only, half for PC2, 25%. Okay, so run this cell. This should ring some bells from way back at the beginning when we talked about ChatGPT and how neural nets kind of look in three-dimensional vector space, because they talked about how do you predict the next word. This is almost a toy version of that. But this is basically has arrows pointing in the principal component direction that each of these x variables is being explained by. So, right, gene A and gene B, really, gene B is almost perfectly in the same direction of PC1. Gene A may just have slightly more variation or something like that, and explained a little bit more by PC2. And this is the kind of plot. The reason to throw it in there is sometimes this makes interpretation easier, because now you can see, well, stress markers going, like, whatever the stress marker is making the enzyme activity go down. At the same time, gene A and gene B are going up, or gene A and gene B going up lead to higher enzyme activity. So, it's hard to pretend on totally cartoon data, but then now we get into like generally looking at the loadings. So this is another way to look at it. I had the table earlier, but you can view that as a bar chart. So the same thing we saw in the loadings. And then the question is, are 2 PCAs the right number? If the 5-way does better, well, what if we simulate, or not simulate, what if we calculate 3 principal components? So now we've got 1 through 3, and this is always the case. If it has to be explaining less variance than the first 2. If it's not, you're doing something wrong. At each stage, it's explaining what variance is left. So now we're plotting PC 1, 2, and 3. So this is where I said you can look at 4 dimensions. So 1 and 2 are on the horizontal, 3 is on the vertical, and now we've got sort of this cloud of points with the colors associated with enzyme activity. So principal component 3, the vertical axis, there's not a lot of color shift along the vertical axis here, right? Sort of similar to the biplot where there's not a lot of color shift across PC2, also not a lot of color shift across PC3. But there's some, so if we did a regression with all three of them, you should explain more of the variance. Oh, this is just same plot, but now with those arrows. Okay. So again, if the thing we really care about is the color of the dot, things that are most similar on the axis where color changes the most, so like gene A is really on an axis where color is changing. And then the really important question at the end of the day, how do you decide whether to use dimensionality reduction? So should we just go with the 5-feature ordinary least squares regression here? Or should we do regression on the principal components? How are you going to decide? What do you want to know? This is the point where we get to the same thing as the gradient descent. Did gradient descent do a good job? Did our approach work? How are you going to justify to your PI that you just ran the 5-way instead of running it on doing principal component to get the dimensions reduced?
I would look at metrics such as R squared, like we were looking at before, and RMSE, and then compare those between the two.
Yep. So which model explained the most variation and had the least residual error in it, okay, or unexplained error. In this case, the 5-way does, so maybe we should just stick with the 5-way. But what about the 4-way? Well, maybe the 4-way would have been better. And then once you get to 5-way, or 5 principal component, then you might as well be doing the multivariate ordinary least squares. So, much like that plot that I had you turn in for the homework, or I'm pretending like it's past tense, it's due tomorrow now. Like, what do you need to look at to convince me that your dimensionality reduction was not necessary? So, going by Zainab's, explanation, I would do the 5-way, right? Because that had the highest R squared and the lowest RMSE. But that's not so hard to imagine or actually compute when you've only got 5 variables, okay? And it becomes a question of how do you decide if I've got 10 or 50 or 100 variables? When do I start needing to do dimensionality reduction? And so these are the plots that you should look at. So what the scree plot, that's the proportion of the variance that is explained by each principal component. So somebody summarize what that graph tells you.
Principal component 1 is explaining like about 45% of the variance within the data, and then subsequently less for each principal component.
Yep. And so by the time you get to principal component 5, you're like 2% of the data. So that doing 5 principal components is almost worthless in terms of predictive power or explaining any of the variation in your data. Another way to look at something similar is it's almost like the flip of that graph is the cumulative explained variance is just summing it up. So after 4, so K of 4, you have 98.3% of the variation explained. 5 at most could explain 1.7%. Now, I only walked through the first 3 principal components and then running a regression on those, right? There was still an extra 11.6% of the variance unexplained by those principal components. And that's why the 5-way is better, because the 5-way is accounting for that last 12% of the variance, okay? Easy to imagine on 5 variables. Next cell, we're going to simulate 200 samples on 100 variables. So think of it as 100 genes. We've got 200 patients and 100 genes. That's one way to think about it. So you simulate that cell. It should do the simulation, and then it will give you those 2 plus one more graphic to look at. Hide my code here. Can't. I need to put time. Okay. So, same kind of plot, scree plot. Somebody interpret this for me. What do you see? I didn't hear you. Yeah, after 4, it's reduced significantly. Another way to say the same thing is almost all of the variation is explained by the first 3 principal components, right? And so, should I do dimensionality reduction before my regression analysis here? Or should I run a 100-way multivariate regression? Okay, I see a lot of people shaking their heads appropriately, right? But then the PI says, why not? Why? And what are you going to do instead? Should I do 50? 20? I want to be able to predict. We can say we've got 100 genes and they're all trying to measure enzyme activity. I want to be able to predict enzyme activity from these 100 genes. What should my regression be? What's the next step here? Feel free to talk amongst yourselves. No guesses about what you would do next? I find this actually sort of fascinating because it seems very clear to me. Yeah, that may be the problem. Should I, what would you use as a model to predict enzyme activity based on this data set?
Yeah, just the first, right?
Yes. Okay. Yeah, I would run a regression on PC1, PC2, PC3 as my predictor variables. What about how would you go about interpreting what that meant biologically?
You have to like figure out what those PCs represent.
Yep. And how do I go about figuring out what those PCs meant?
Do that little table that you did earlier.
The loadings, yeah. So, or a graph like this that shows the loading on the genes. And then, right, this is where working on a model system makes it much easier than working on a non-model system, because you might know, instead of having this laundry list of gene 1 to gene 100, you've actually got names on those genes with functions on those genes. So you can say all these genes that have this functional annotation load really high on PC1, okay? And PC1 has this relationship after I run my regression on the top 3 PCs, right? I'm looking at which genes load highly on PC1, okay? And PC2 and PC3, okay? The last one there is introducing a new concept, the data partitioning, okay? The word is escaping me now. It's not co-validated. Cross-validated. Thank you. Good grief. That was a total brain cramp. Right. So this is 5-fold cross-validated R squared. So what this is it partitioned the data into 5 subsections and reran the model each time. So basically showing you the amount of variation explained and how consistent that is depending on how many principal components you include. And so you maximize it after 3, and then after a while that cross-validated, like how consistent is it, because because of the correlations and how many genes, how many predictor variables you have. Once you're out here, they're all sort of explaining the same variation, and so the R squared is not consistently improving. Okay, that's one way to think about it. It's a super useful plot to, like, another way to tell you, because it's not always this clear, right? There's a giant drop off here in between 3 and 4, but it's not always that obvious. So this is another plot that will tell you where the inflection point is, potentially. So you can say, justify or rationalize why you pick the number of principal components, like how reduced is your dimensionality. And then last thing is if we want to visualize these principal components, what's a good way to do that? So the last cell will compute a UMAP and a t-SNE plot. So this is a 2-dimensional projection of all of those 100 variables on sort of like two principal components, except this is a non-linear relationship now. So I've got a couple slides in here, like just talking about, these are both designed to show you how these things, how your enzyme activity in our example, how your Y variable, the thing you're trying to predict, are related across all of the variables. And so the way that they do that conceptually is basically take a distance matrix, giant distance matrix of all of the samples from each other, like how different is gene 1 in all the samples from gene 2 and so on. So you've got a giant distance matrix, and then project that as best you can into 2 dimensions. So what it does is if you have a very similar, say, so think of it like you've got 2 genes that are super highly correlated, you want to plot those next to each other in 2-dimensional space, okay? So that's really all it's doing, a super complicated version of that, right? It's trying to take that whole distance matrix and put everything that is most similar close to each other in 2-dimensional space. And the difference between UMAP and t-SNE is just what do they do to try to maintain global structure? So t-SNE doesn't do anything to try to maintain global structure. It just does that sort of first pass of it's keeping things that have high correlation close together. UMAP tries, it does that hierarchically so that you have, think of phylogenetic trees. We've seen some hierarchical clustering when we did heat maps. So that, think of those trees, there's more than just the similarity in the tips. So different levels of organization will get pulled closer or farther apart. So think of it like a deep branch in a phylogeny will be far apart in a UMAP plot, okay? And so it's trying to differentiate the clusters by maintaining some kind of global correlation structure. In the case of this simulated data, there's not a ton of global structure, but you can see how it's a little bit more spread out in mine, and I don't remember if I left it random. Does your picture look like that, or is it different? Yeah, I think I left a random seed. There's a weird thing in the function for UMAP, this is totally in the weeds, but if you set the seed, it does it serially, so it only uses 1 CPU, and it takes much longer. If you leave it random, it will parallelize it and figure out different parts of the map in parallel. And so I left it on random just so it'd run real fast. I don't know. I might have. Oh yeah. So I left it in there. You line 28 and 29. You can switch which line is commented out there. And so what you'll see is if you rerun this the way I have it set, it will change the picture slightly every time. But if you switch it so that the random state equals 42, and 42 is an arbitrary number, it just starts with the same random state every time, it will give you a warning, which is how I found out about this problem. It doesn't do anything, like it still runs fine. That warning is not saying there's something wrong with your analysis. It's just saying this is going to take a long time because you're running on 1 CPU. Okay. Questions? We are done with dimensionality reduction in some sense. Very dipped our toe in the water of nonlinear projection for those dimensions in t-SNE and UMAP. Thursday we will say Okay, we've done that dimensionality reduction and projection. Now, can we classify things based on that? So we will talk about support vector machines, where we know based on some markers that the little cluster over here is because gene A and gene B are super downregulated. Okay, now predict what kind of cells those are, or what kinds of samples those come from. Okay, that's it for today. If you haven't turned in the homework, that's due tomorrow.
I'm still not wanting to. I've changed this like so many times. I've realized I submitted the bottom one and not the top, so I like updated it. But I've changed it so many times for it to curve at 20,000, but it won't do like afterwards. I don't know where I'm going.
At 2,000 or 20,000?
2,000, but I don't know where I'm going wrong here. So I even have it here. It's like 2,000. I don't know where I'm going wrong.
So where does it set there? So that's ETA. So
Should I?
yeah.
I think I put an extra 0. Is that the problem?
Yep.
Yeah.
So if you take that off, that you're gonna see what you were expecting to see.
Okay.
Just rerun that cell now.
Oh, wait, I think I put it here too. Oh, I don't know. Let's see. No? What the heck?
Yeah. So what do you do to make it go slower?
I need to change the ETA.
Mm-hmm.
Okay. Alright. I feel better.
You got it.
And then to remove my old graph that I turned in on Friday,
You don't have to remove it. You
Okay.
just by default it only shows us the most recent submission.
okay.